So, Lorenzo Galluzzi is one of the most dynamic scientists that I know. He has multiple appointments in USA, France, and Italy, including Weill Cornell Medical College, Yale School of Medicine, University of Ferrara, University of Padova, and University of Rome, La Sapienza. He's also associated director of the European Academy for Tumor Immunology and founding member of the European Research Institute for Integrated Cellular Pathology. He is the recipient of so many awards and honors, and I am not gonna through of details for the sake of time. Suffice to say that Lorenzo has made extremely important contributions to the fields of cell death, autophagy, tumor metabolism, and tumor immunology by providing insights into the links between adaptive stress responses in cancer cells and the activation of a clinically relevant tumor targeting immune response. I feel very happy and honored to have Lorenzo again in our course. And Lorenzo, I hope the next time we will have you here in person.
Thank you very much, Karina, and thank you very much, Gustavo, for the invite. I also hope to be there in person next time. Absolutely. I really enjoy being here with you today to share some of the concepts that we developed over time. I'm going to try to share my screen, and you guys tell me if you actually, I'm seeing that I'm disabled to share. Can you enable me?
Márcio, can you...
Márcio, deixa ele compartilhar a tela.
Pronto. Now.
Okay.
Now you can.
Yeah, now it seems to work. Just tell me if you see my slides and my screen.
Yes, yes, that's okay.
Okay, so I'm happy to be here and to answer positively the invitation from Gustavo and Karina to try to go through a little bit of basic cell death concepts and why there seem to be so many ways through which the cells are dying. This is something that we care a lot in the cell death community because there's often a misinterpretation of what's happening with the cells and what is the outcome of cell death and what are the mechanisms, the actual mechanisms that drive cell death. So these are my disclosure. I happen to be consulting or scientific advisory board member of a few companies, but nothing of what I'm presenting today has anything to do with this work. So when you think about cells that are exposed to a stress condition, as when you receive this kind of messages on your cell phone, the cells are actually stimulated and they need to respond as you need to do if you receive these kind of messages. So the cells are tending to panic as you do, and to recover homeostasis, to try to adapt to this stressful condition, they use intracellular adaptation mechanisms. Those mechanisms are there to preserve the homeostasis of the cells. They have been evolving throughout evolution from very simple mechanisms to more complicated pathway that I will describe a little bit more in details later. But the idea here is there is a first attempt of the cells to save themselves and to return to their physiological role. And this applies not only to mammalian cells and human cells, but it applies to unicellular eukaryotic organisms too. So if they are, the cells are capable of restoring homeostasis because maybe they receive some positive feedback from the environment, as in the case of this nice drink here, then the physiological function of the cells are going to come back up. The cells will survive, and of course the organism will be happy because at the end of the day, the identity and function of that specific cell has been maintained. When this does not happen, there's two different terminal fates that the cells can undergo. One is called cellular senescence, and the other one is called regulated cell death. We called it regulated cell death, and I'm going to explain you in a second what it is. But the idea is that in this case, homeostasis is for the cell is irremediably compromised. So the cells are unable to go back to their functions. They are unable to go back to serve the organism in the way they should have if it's a multicellular organism. And of course, they will succumb if it's a unicellular eukaryotic. So what is regulated cell death? Regulated cell death is a concept that we introduced to describe a type of cell death that is mechanistically determined. There's a genetically encoded apparatus that is there to regulate the way through which the cells are dying. And I'm not saying to kill the cells, which is different, but to regulate the way they are dying, which is a bit more complicated. It stands at opposite to another type of cell death, which is called accidental cell death. In that case, there is no mechanism. In that case, it's simply a strong, very strong stress condition that will tear the cells apart. If you take a flame, for instance, and you burn your cells with the flame, there's no genetically encoded pathway coming up. The cells will burst apart. Or if you put the cells in pure water, the cells will just break into pieces, and it is a non-regulated process. On the contrary, when you expose cells to a stress that is beyond the adaptive capacity, so cells are unable to recover homeostasis, but they die trying to recover homeostasis. In that case, there is a mechanism that comes in place. And here I also depicted a variant of regulated cell death, which we call programmed cell death. And that is a concept that applies to strictly physiological scenarios. There is no stress in that case. The cells are dying all the time in your body just to preserve your body function, just to preserve your body identity. Those are not stress cells. And as the same is during embryonic development, cells are dying because they need to die to shape, for instance, your hands and to make the interdigital space. There's no stress involved there. The cells are dying because there's a program that tells them you need to die now for the sake of the organism being in good shape and being functional. On the contrary, when there's a stress, so that's another story. The cells are responding and they're trying to recover homeostasis. And when they cannot, they undergo regulated cell death. So we should take another step back, actually, and to ask ourselves, when can we define a cell as dead cell? It's not even easy sometimes to define death for humans, as you probably know from like these journal reports and like the newspaper saying that the guy who was in a casket and they were doing the funeral service, at some stage, he just stood up and left the casket. So it's even more complicated for cells because unfortunately you cannot see that on this kind of figures under the microscope. So you need to define something that you can measure, something that is irreversible, because at the end of the day, the concept of death is in its irreversibility. If a cells can go back to their function, they were not dead, whatever some people will try to convince you about. The same thing goes for people. If a people is in a casket and can get out of it and then walk, that person was not absolutely dead. So there has been a lot of attempts over the years, actually over the past 3 decades, to define this point of no return, this moment where whatever connects cell life to cell death becomes irreversible. And of course, the idea was to try to put it as upstream as possible in the cascade of event. And many processes have been proposed, and I will suggest a few later, to define that transition, to define where the life is irrecuperable, and so the death comes in. But from a very pragmatic perspective, this is very complicated. So the cell death community agreed already 15 years ago to focus on events that are really irreversible. And those are the complete loss of plasma membrane integrity and cell fragmentation. When the plasma membrane has holes in it, the cells, and those holes are big enough and there's enough of them, the cells will never be able to fix it, and they will lose their homeostasis. Remember one thing, when we think about evolution and we think about homeostasis, the first feature for homeostasis is the capacity to control a physically defined space. You cannot maintain homeostasis if you cannot define a space where you can do chemical reactions that are supporting this homeostasis at the expense of chemical reaction outside of that space that you do not care about. This is a thermodynamic principle. You cannot maintain organization in a space that is not physically defined. So this goes down to thermodynamics. If the cells has broken membranes, they cannot maintain homeostasis in there. And that's where we now try to really focus when we measure cell death. And I'm not saying that there's not other mechanisms that drive that or that regulate that. I'm saying that this is when we should consider a cell as a dead one. So this is a happy cell. It's a cell that is alive and that has a intact plasma membrane. Some cells activate caspases, which is one of those processes that I mentioned before has been suggested as a way to define cell death. And indeed, caspases are, in some cases, linked to the activation of a cell death mechanism, as probably you've heard earlier in this course. But until the cells have an intact plasma membrane, that cell is still a living cell. It doesn't matter if caspases are activated. Most likely, if caspases are activated beyond a certain threshold, yeah, that cells will die, but at that time where the plasma membrane is intact, we still consider that as a living cell. Why this is important? This is important because caspases do not only kill cells, caspases are also involved in many non-lethal processes. For instance, neuronal differentiation. So if you have a neuronal progenitor in your brain that needs to become a neuron, that progenitor will activate caspase 3 at specific location in its cellular compartments, and that caspase 3 will be important for the neuron, for the precursors to become a neuron. So if you define cell death by caspase 3 activation, and you go to the brain, maybe what you're looking at is not cell death, it's just neuronal differentiation. And the same is for autophagy. Autophagy is a more complicated concept that I'm going to explain a bit later here, but there has been a lot of, in the past, there's been a lot of investigators thinking that autophagy would be a cell death mechanism. In most cases, it is not. We can go, we're going to talk about autophagy a lot later. But even if it were, even when it is, because in some cases it is, cells are not dead until their plasma membrane is not permeabilized. So now you have a living cell. Here you have a living cell with caspase activation, and you have a living cell with autophagy. If those cells get their membrane permeabilized, now they're dead, together or because of caspase activation, together or because of autophagy. And listen carefully here, together and because is very different, and I'll tell you later why. So why is this so important to define cell death in a strict manner? And I'm going to use an example, which is my marriage, by my failed marriage. Actually, I was married to this beautiful girl back in the days, and we were happy for a long time, but at some stage we decided that it was not good enough. And actually, I think it was my fault, because if I had done a very, very strict literature search, I would have found that in 100% of the cases, divorce is preceded by marriage. So I was putting myself at a risk by marrying this girl to have a divorce. And some people would actually interpret that by saying that weddings and marriage causes divorce, which is something that is a little bit more controversial. Why I'm telling you that? Because when you think about regulated cell death, as I mentioned before, regulated cell death is preceded by an attempt of the cells to restore homeostasis in 100% of the time. When you stress your cells, they will try to adapt. If they cannot adapt, they will die. But based on what I just mentioned, some people are thinking that adaptation to stress is the cause of cell death. That is why I was putting this emphasis on together or because. And this is something that we can discuss, and we will discuss a lot here. And nonetheless, as I mentioned before, if you go to the books, the textbooks, many of those that you are studying for university, and you go to the chapter on cell death, you will find this old school notion that cell death can be classified in 3 types, which is apoptosis, necroptosis, and autophagy. Where does this come from? This comes from the 60s and the 70s, where the only thing we had to study cell death was a microscope. And people were looking into the microscope, they were looking at cells that were dying, and they identified morphological features of dying cells. So for apoptosis, for instance, they were seeing that the chromatin is getting condensed, that the nucleus is fragmenting, that the cell itself is shrinking. For necrosis, they were seeing none of this. They were seeing actually some vacuoles and some swelling of the cells. And for autophagy, they were seeing a lot of these vacuoles. But the problem was they didn't have the tools to understand if what they were seeing was actually the cause of death or was a phenomenon that was accompanying that, and it was unrelated, or it was even perhaps a phenomenon that was trying to avoid that. And yeah, and this is how we feel today about that classification, I believe, across the cell death field. So going back to autophagy, which I mentioned before, and some people believe it's a cell death type, autophagy is actually a very old mechanism through which cells try to respond to stress. It's a cytoprotective mechanism. There are several variants of that. I will talk about macroautophagy for the rest of this talk, but there's others. One is called chaperone-mediated autophagy. The other one is called microautophagy. And they different mechanisms, but they all have the same purpose. They all have the purpose of clearing the cytosol of the cells from trash. Autophagy is there to pick up trash, to pick up disposable organelles, to pick up malfunctional organelles, to pick up invading pathogen or virus species, and to segregate these species in an organelle that is called autophagosome, which is represented here at the center. And that organelle fuses with lysosomes to degrade the content. And all this trash here can be very detrimental for the cell, extraordinarily detrimental, as much as the genes involved in autophagy are all required for embryonic development. If you don't have one of these genes in your mouse, then you don't have a mouse. They all die shortly after or shortly before birth. They are even adult animals where you can use now new technology to delete a gene in a mouse that is completely formed. If you delete one of these genes, they will die. So autophagy is as important as that for the preservation of homeostasis and for organismal development. And this is another point about what I said before. It is very different to observe two things at the same time, as it was done for autophagy and cell death, as to believe that they are connected etiologically speaking and causally speaking. And this is just a plot of people drowning because they fell in a swimming pool and per year, and also the movies where Nicolas Cage was starring. And I don't believe that people were suiciding in a pool because of the Nicolas Cage's movies, or that Nicolas Cage was doing movies because people were falling into pools, right? So we should really be attentive when you draw a link between two phenomena and you think it's a causation link. Why, again, it's so important to define cell death in a strict manner. And I took this as an example from the real scientific world, not jokes anymore. This is an assay that people use to test the sensitivity of cancer cells to a drug that is called bortezomib. Doesn't matter, could be any drug. This assay is a commercial assay that is making some big pharma very rich, and I'm happy for them, but there's a problem here. So this assay is based on the assumption that you can measure cell death by measuring the content of the ATP in your cell culture. And of course, when your cells are happy, like in the blue curve, there's no problem. You assume that this is the normal amount of ATP they should have, and it's a perfect scenario. Your cells are happy and kicking. Also, I don't have lots of problems when the cells have no ATPs, because I mean, if you have 0% of the ATP content of your control condition, it most likely means that all the cells are dead. So I'm very happy with what happens in the blue curve here, and very happy with what happens in the green or purple curve here. What I'm unhappy about is what happened here. So here, the graph tells you you have 80% or 75%, whatever that is, of ATP in your control, of your control condition. And this paper is saying, okay, this means I have 25% of cell death. My question is, does it really? For me, it would also potentially mean that I have 0% cell death, but each of the cells now contain 75% of ATP as compared to the initial condition. 75% of ATP is perfectly compatible with life. There's no problem. Maybe these cells have just exhausted their culture medium and they need more food to get more ATP, but they're not dead. So this is a very dramatic point here, because my interpretation is there's no cell death, and the interpretation of these people is 1/4 of the cells are dead. And that's why it's important to define cell death in a way that is absolutely unquestionable, which is the integrity of the plasma membrane, as I mentioned before. And this is why I spend every 3-4 years, I spend 2 months of my time organizing a committee of people that has something to say about this kind of definition, and has something to say about how we should measure stuff in the cell death community, and how we should interpret what we see in our papers, as well as in the papers from other fields. So going back to cell death, and this is, it speaks a little bit of what we just said before. So cell death is a complex thing, okay? It has different dimensions, it has different aspects. One aspect is morphology, and we discussed this a little bit before. One aspect is biochemistry, and one aspect is outcome. But as you can see here, these dimensions are not on the same axis. They can vary in differential manners. In the past, it was considered as if they were varying along the same dimension. In the past, we would say, okay, apoptosis as this morphology is always immunologically silent, so there's no outcome in the organisms, and it's always driven by caspases. And necrosis is never driven by caspases, is always this morphology, and is always immunologically inflammatory or active. Now we know it's not the case. Now we know it's more complicated than that. Now we know, for instance, that there are instances of apoptosis that can be immunologically active. They can drive an immune response, even though they have the same biochemistry that we define for apoptosis, and even though they have the same morphology that we define for apoptosis. So this is what I just mentioned, and this is the old school. So look into a microscope, in this case, an electron microscope, but you can also probably do part of it in a normal microscope, and you see how these two things are different. And that's where in the 70s, somebody said, okay, we call this apoptosis, shrinkage of the cell, nuclear condensation and fragmentation, very clear, very dense electron cytoplasm. And we call this necrosis, something that is basically exploding, swelling, mitochondria are swollen, the nucleus is swollen, and another one down here. This is beautiful, and everybody who's listening can probably, if I send you another picture of those, can probably tell me which is apoptosis and which is necrosis, and that's amazing. The problem is, when you look at the microscope, sometimes you find this, sometimes you find this on the right, but most cases you just find something that is intermediate. So now you have a beautiful swollen cytoplasm, but you also have nuclear condensation. So now how do we call that? Necroapoptosis, necrosis, how do we do? When is the cell death, as all of the biological disciplines, is not a place where there's a strong dichotomy. There's always a spectrum of phenotypes, especially in the morphology scenario. So this is a morphological spectrum, and it's very hard to put a bar and say whatever is on this side is necrosis, whatever on this side is apoptosis. So what we did as a cell death community, we dropped it. We don't call cell death apoptosis and necrosis anymore based on morphology. We introduced a better way to do it, which is mechanistic. Now we don't care anymore what the cells look like. I mean, we care, but we don't use it as a definition. Now we define cell death based on something that we can measure with a machine, which is much more reliable than measuring it by your own eyes as you count the cells that look one way or the other way. And more importantly, that we can modulate pharmacologically or genetically. And this is just a very, very simplified scheme about some of the types of cell death that we have now defined in this manner. But to make you a very complicated thing much simpler, this is apoptosis. There's two pathways. One originates outside of the cells, the other originates inside of the cells. And the definition is that we can slow it down, we can partially inhibit it when we inhibit caspases. So now you are in a setting where you measure cell death with your very potent approach that you measure plasma membrane integrity, which is very simple. There are molecules that can go inside the cells only when the plasma membrane is broken, and we call those vital dyes. One of those is called propidium iodide. So assuming you're using a drug, and one day later you get 50% of your cells that are propidium iodide positive, or they have plasma membrane that is permeabilized, admit also that you tried to inhibit that or to change that with a chemical inhibitor of caspases, and indeed 24 hours later you only see 25% of those cells with a broken membrane. Now you can say, okay, the mechanisms that is at play here is apoptotic cell death. Why? Because I can delay it, I can post-dissipate it, I can slow it down by inhibiting caspases. And there's other ways. There's many ways of regulated necrosis. We call it regulated necrosis because it looks like necrosis when you look at it in the microscope, but we define it not by looking at the microscope, but by their dependence on some of the proteins that are indicated here. So now we have a biochemical, reproducible way to define cell death, which is much less prone to interpretation. Machines are machines; they give you a number. If the numbers are that, they're gonna be the same here, probably the same in Sao Paulo or in Brasilia and in Europe, if the experiment is properly performed. So that biochemical definition of cell death brought about a lot of new nomenclature. When I started this field, and I was a kid in 2005, yeah, we kind of had 3 or 4 of this. We had intrinsic apoptosis, we had extrinsic apoptosis, we have so-called mitochondrial permeability transition, and basically that was it. Necroptosis, ferroptosis, pyroptosis, but all these guys came about later. Why? Because we started to study the mechanisms of cell death in more detail and more details, and now we identified, for instance, this regulated version of necrosis I mentioned before, which is called necroptosis. And it's been really a personal pleasure to see the field evolve so much and to see all these pathways identified, because when I was at university at the end of the 90s, I was also studying that apoptosis is regulated and necrosis is not. And now we know it's not as simple as that. Another thing came up, and I think it's equally important here. According to our previous understanding of cell death, we knew there were different ways to kill cells, right? And but we also thought that they would be not interconnected, so that they would proceed along tracks that were completely non-overlapping and parallel to each other. So with a specific lethal trigger, you would get this type of cell death. With another lethal trigger, you will get another type of cell death, and with a third lethal trigger, you will get a third type. This kind of model would predict that if you can inhibit a specific pathway of cell death, then you're going to be able to save your cells from that specific lethal stimulus, which it turns out it's not the case. Why is it not the case? Because we know now that these cell death modes are communicating to each other. When a cell death mode is engaged, it's not engaged on a parallel track that is completely, that the cell is completely unaware of. No, the rest of the cell is talking to the cell death pathway. So now, even if you inhibit that specific cell death mode, probably you're still gonna have cell death, maybe with a different kinetics, maybe with a different morphological manifestation, maybe with a different immunological manifestation. But most likely, if the stimulus was strong enough, you're still going to get cell death at some stage. Because this, as I will tell you later, these mechanisms here are mostly in place to regulate the speed at which the cells are dying and the immunological signals that they release when they die. They are much less there to decide whether a cell is dying or not once the point of no return that I mentioned before, that actually nobody knows where it stands, has been passed. So there is a different, I think we start to think about all these mechanisms in a different manner, also because we started to investigate these processes not only in plates by using cells that have no microenvironment, but by using model organisms to study cell death in the organism and to see how the organism is responding to that. So, and I'm going back a little bit to what I said before because there really is a better, like a more refined appreciation of what is important and what it is not important for the cell death to happen. So, as I told you before, when you have this relatively mild homeostatic perturbation, which is a nice combination of words to say stress, then your cells are trying to adapt. And if they succeed, they're going to recover homeostasis, as simple as that. This you can pharmacologically modulate. You can make this better. You can make the adaptive response better. You can save cells if you want until you are talking about this early pathway, until the cells have not committed to die. It's a pre-cell death scenario. It's not cell death yet. It's a response to stress. And this is very modulatable from a pharmacological perspective. For instance, you can boost autophagy. If you boost autophagy, autophagy is very important here, then you can make your cell more resistant to stress. When cells are unable to cope with stress, then they go through this point of no return. And I told you, nobody really knows what it is. And then this point of no return will cause two things. It will cause the driver of cell death, the actual mechanistic link between failing stress and cell death, but it will also cause other stuff that are around that you can measure, but they don't influence the speed and the progression between the point of no return and cell death. And we call those correlates because, yeah, they are there, but they don't matter. They're just there because, you know, it's biochemistry. Things are happening in the cells. If you change something, a lot of things are changing. Doesn't really mean they are connected to the pathway. What do we believe these causes are? I think there's more and more appreciation to the fact that these causes are two. Cells are dying because they lose energy. So they lose ATP. They are unable to preserve homeostasis because the preservation of homeostasis requires energy. Doesn't only require a confined microenvironment. It also requires energy. No energy, no preservation of homeostasis. And because of the structural derangements that are caused by oxidation. One of the first things that happens in the cells when they are going through this point of no return is the loss of mitochondrial function. And that means no more ATP or much less ATP, and it means overproduction of reactive oxygen species. And both these things are targeting the very core definition of homeostasis, which is a microenvironment and an energy source. And then, of course, there's these correlates around it, which I'm going to talk a bit more now. In this case, it's basically the same stuff, but there's no adaptation. In this case, you do directly, when it's something very strong, as in the case of accidental cell death, you directly go to death. And this is difficult to prevent. It's not pharmacologically modulatable up front. And it's complicated in this setting either. What happens now? As I mentioned, the cells are not really dying in a plate. They are dying in a microenvironment where there's other cells. And the outcome of this cell death here that can be regulated or accidental, depending if there was an adaptive response or not. So these cells are dying. They are losing the membrane integrity. So they are releasing things. They are releasing things that are bioactive. Those things can be cytotoxic molecules, for instance, reactive oxygen species, or so-called damage-associated molecular patterns. Those are danger signals. Those are the very way through which dying cells talk to the organism, talk especially to the immune system, which is there to control whatever instance of cell death is around. So these agents here, as I mentioned, the cytotoxic one can cause additional cell death, and they both can regulate inflammation basically and immunity. In either way, it can be, they can activate immunity or they can suppress immunity depending on what was the cause for cell death. If cell death was part of a physiological mechanism that is in place to preserve the organism without reference to stress, for instance, the turnover of your intestinal epithelium, then there's going to be an inhibition of inflammation, because you don't want your organism to mount an inflammatory response or an immune response toward your healthy intestine. You don't want that. On the contrary, if this perturbation of homeostasis was a viral infection that ultimately killed the cells, then these damps here, these danger signals, will activate an immune response, because now you want your immune system to take care of those dying cells and the cells nearby that have a viral infection too. And this is critical for the preservation of organismal homeostasis. But as you see, it's two differential concepts. In this case, the organism preserves homeostasis by preserving the homeostasis of the cell if it is possible. If it is not possible, the organism preserves homeostasis by sacrificing the cells and controlling the outcome of that cell death. So now the cells are serving the organisms, are no longer serving only themselves. And that's where we came back in 2005 with this definition of immunogenic cell death. What is immunogenic cell death? It's a type of cell death that doesn't need anything else to drive an immune response, and not just inflammation. Here we're talking about an antigen-specific adaptive immune response with CD4 and CD8 T cells, not just macrophages, not just NK cells, not just inflammation, but really an antigen-specific immune response. How does this work? If the cells are killed in the right manner, and I'll tell you in a second what the right manner means, they're going to be able to attract immune components to the microenvironment. These immune components will be activated. They will migrate to the lymph node and they will prime T cells specifically for antigens that were expressed by dying cells. These cells will go into the circulation and they will be able to go back to the site of cell death, perhaps killing cells that are resistant to therapy, and they will generate immunological memory. And so what is the best way to test if immunogenic cell death, if a cell death type is immunogenic? You kill your cells in vitro and you use them as a vaccine. And if that is protecting against an injection of the same cell later, of course, we're talking about cancer cells here, then you have immunogenic cell death. If not, then your cell death was not good enough at doing this trick, at doing what the vaccines are doing. And it's funny because Guido and myself, back in 2012, published this nice review on immunogenic cell death, and it was very, very nicely accepted by the field, but probably not as much as the so-called cancer immunity cycle that Chen and Melmal put out a few months later. And you can see the resemblance here. There is a release of antigens, the activation of an immune response, and then the immune response can go back to the microenvironment to kill residual cells, in this case, these cancer cells, and the same happens here. What are the ingredients of immunogenic cell death? It's not so easy as it looks like, because as I mentioned before, you don't want your normal cells when they die, because they need to die to preserve your integrity, to elicit an immune response. So you need multiple ingredients. If one of these ingredients is not around, then there's no immunogenic cell death. And I think most of the examples I'm going to do are related to cancer from now on, but there's also cancer unrelated instances of immunogenic cell death. And I will probably allude of some of them over the rest of my talk. So first of all, you need cell death, okay? Because if you don't have cell death, you cannot have immunogenic cell death, and then it's relatively simple as a concept, right? And so you need absolutely cells to be dying. Why? Because living cells are not good food for dendritic cells. They are not good food for macrophages. So they cannot be processed towards antigen processing and presentation. So you need cell fragments, you need cells that are dying. If cells are dying, but they don't have antigens, and I'm going to come back to this in a second, they only release these DAMPs that I told you before. But in the absence of antigens, these DAMPs are going to generate an inflammatory reaction, but no adaptive immunity, no CD8 positive, CD4 positive T cells, nothing that is antigen specific. And this inflammatory reaction over time is going to fade away unless cell death continues. And then in that case, you will have an inflammatory disease. But it's not, again, adaptive immunity. It's just innate immunity. So antigens are very important here. And they need to be antigens that you have not been tolerant to. As you know, your CD8 positive and CD4 positive T cell compartment is informed, is educated in your thymus to only recognize foreign antigens, or they called it in the past If you think about pathogens, then it's easy. If your cells are infected by a virus, the virus encodes proteins that will generate antigens. That your body has never seen those antigens when you were developed the T cell repertoire. So then you have a source of antigens that is available for driving an antigen-specific immune response. But it's not only the case. Cancer cells, for instance, are mutations. Mutations can generate antigens that were not there when your circulating T cell repertoire was developed. So also cancer cells can be potentially recognized by T cells. And there's another category of antigens that I like very much, which is less investigated, less understood, but I think very important in many different diseases, which is post-translational antigens. As you know, your proteins are encoded by the genome, and probably that version of the protein was in your thymus, and so the T cells that recognize that were deleted. So you cannot recognize the normal or original version of the protein. But what happens if the protein is oxidized? For instance, because you have inflammatory reaction. If the protein is oxidized, then it will be processed in a different manner by the antigen processing compartment, and it might get exposed on MHC molecule on the plasma membrane. And now you have a different protein because the oxidation changed it. And then maybe you have T cells that can respond to that. But anyway, so you need antigenicity. But funny enough, if cells are dying with antigens, but they don't produce these DAMPs, actually, you're going to get the contrary. You're going to get the deletion of the T cells that need to, that are specific for those antigens. So you're going to get tolerance here. What you really need is both these antigens and the DAMPs. And now finally, your system will be able to prime an immune response against those specific antigens that are expressed, in this case, by cancer cells. This is only priming, though. This means that the immune system was able to pick up antigens, go to the lymph node, and generate an immune response that goes into circulation. However, these T cells still need to get back to the tumor. So if the tumor is protected by the microenvironment, you will not get the tumor killing by the T cells that you have generated. So you also need a permissive microenvironment. You also need a microenvironment that allows T cells to go in and to exert their cytotoxic functions. Why do we believe this is so important? Because if you look at pathogens, and if you look at cancer cells, you will see that they evolved many, many different ways to avoid it. If it was not important, pathogens would not care. But both pathogens and cancer cells, they evolved strategy to, for instance, resist cell death. So when you had a viral infection at the very beginning of the infection, the cell will be protected from dying because the pathogens need to avoid this activation of immunogenic cell death. And many pathogens and cancer cells, they're able to target specifically those signals that I mentioned, and there's a few of them. One, for instance, is extracellular ATP. When dying cells are producing a lot of ATP, that's immunostimulatory. So what happens? Cancer cells overexpress an enzyme that degrades extracellular ATP to avoid an immune response. And of course, there are activities that can be used to circumvent that. So we can therapeutically go after these problems. We can therapeutically say, okay, there is a problem at a certain level. I can use a strategy to circumvent it. Another good example here is CD47 upregulation. CD47 is a molecule that prevents phagocytosis. So when cancer cells are dying and they express a lot of CD47, they will not be taken up by dendritic cells. They will not be taken up by these cells that are needed to drive an immune response. So what can we do? We can block CD47 with some antibody. And I want to put again the attention to autophagy here. Why? Because autophagy here is involved in two different aspects of immunogenic cell death in a diametrically opposed manner. Autophagy is important for cancer cells to release ATP when they die, so it has a beneficial effect on immunogenic cell death by promoting the release of ATP, but autophagy inhibits type 1 interferon secretion, and type 1 interferon is one of those cytokines that are important for immunogenic cell death. So now we have a scenario where autophagy is doing two opposed effects. It's beneficial on one end and it's detrimental on the other end. And I'm going to tell you more about it now. So if you think about autophagy, what I told you at the very beginning of the talk, as part of this activity of protection from a simple cell intrinsic perspective. So cells are using autophagy to survive stress. So from a cancer perspective, you would think of autophagy as a bad thing. Why? Because you want to kill cancer cells. So if autophagy helps cancer cells to survive stress, you want to inhibit autophagy. There's a lot of problems with the inhibitors that are available, but let's forget about that for a second. There are inhibitors. They are non-specific, kind of toxic. The original idea here, which has been tested, and I will show you, was, okay, now we do not only try to kill cancer cells with a treatment that can be radiation or chemotherapy, whatever you want. We want to maximize the number of cancer cells that are dying because we inhibit their capacity of adaptation by blocking autophagy. And I call this the Starbucks approach because it's a quantity thing. You want to increase the number of dying cells. And it kind of works if you do it in a plate, of course, and also if you do it in immunodeficient animals. In this case, immunodeficient mice implanted with colorectal cancer cells that were either wild type or control, or they were inhibited in autophagy because they lack this gene, which is called ATG5. And we irradiate them. When we irradiate the control cells, we get a response, which is here. When we irradiate the autophagy deficient cells, we get a better response, which is here. Not surprising. I told you cancer cells are using autophagy to survive stress. Radiation is a stress that kills them. But if they don't have autophagy, then they cannot count on that mechanism and they're going to die more. So this kind of reasoning has brought so many people to think about, okay, Starbucks is good because now we are able to kill more cancer cells. Let's try it in patients. And there's been clinical trial with that, combining different types of treatment with those drugs I mentioned before, chloroquine and hydroxychloroquine. They are bad drugs, but they can be used in cancer patients. And the summary of this table is very simple. It's relatively safe. I mean, it's not the best in the planet, but cancer patients have a lot of other problems. But it doesn't work. It does not change the activity of the treatment per se. There's no amelioration of the treatment. So there's no added benefit by combining treatment of whatever that is, radiation, drugs, different drugs with autophagy inhibitors. And that is the same. Why? People started to think, why now we can make cells dying more, but we don't get an advantage in the patients? Because of what I told you before, this is not happening in a plate. This is not happening in an immunodeficient host. This is happening in an immunocompetent patient where the help of the immune system is major. And this danger signaling here is impacted by autophagy. So now you don't have autophagy anymore. Maybe you're dying, you're getting more cells to die, but then you compromise all this help from the organism in recovering homeostasis. So we came around, when I was still working with Guido Kremer in Paris, we came around with another approach, which is the other way around. We know the Starbucks approach is not particularly good because, yeah, we kill more cells, but we compromise the immune control, so we still don't see advantages in the patient. Maybe we should try to boost autophagy we know we're going to lose part of the cytotoxic response because we're going to promote some sort of adaptation by cancer cells. But we are also making these few cells that are dying more immunogenic because autophagy, as I mentioned before, is linked to this dampenation. So now maybe the immune system is going to help us out. Yeah, we kill less cells with treatment, but maybe the immune system is going to kick in and kill the rest of the cells. And we're going to have immunological disease eradication. And I call this the espresso approach, because it's no longer a problem of quantity, it's a problem of quality. And we started to seek for data supporting this. And the first thing we did, we generated cells that had, again, autophagy incompetent because they lacked ATG5 or ATG7. We put those cells in immunocompetent now animals, and we treated the animals with a drug that is called mitoxantron, which is an immunogenic cell death inducer. You see here, when the cells were autophagy competent, mitoxantron had an effect. When the cells were autophagy deficient, mitoxantron lost completely its effect, meaning that autophagy was important for the efficacy in cancer cells, was important for the efficacy of mitoxantron. And why that? As I mentioned before, autophagy is important for emitting ATP when cells are dying. And ATP is critical for immunogenic cell death to drive anti-cancer immunity. Here we use the same cells in vivo, but we've also engineered the cells to express a luciferase that would measure extracellular ATP. And what you see here is when you treat control cells, you see an increase in extracellular ATP, which is quantified here. And this does not happen when you treat ATG5 deficient cell. This is the treated mouse, and this is the untreated mouse. You see, you actually see a decrease. And if you use the same cells in the vaccination test I mentioned before, so you really take naive animals and you use cancer cells that have been killed in vitro by a drug as a vaccine, and then 2 weeks later, you give the animals living cells to test whether the animals can reject these cells. When you kill cancer cells in a plate with mitoxantron and you use these dying cells as a vaccine, it works really nice. 80% of the animals are vaccinated, prophylactic vaccination. If the cancer cells you use for the vaccine are lacking genes that are involved in autophagy, then you lose that effect. You get down to 20%. So autophagy is important for the delivery of signals that drive an immunogenic response in the mice. Of course, we wanted to go on all the other way. Now we just proved that inhibition of autophagy was bad. We wanted to prove that activation of autophagy is good instead. And there's many ways of doing it. One way is to just starve your mice. You take your mice and you leave them without food 24 hours once a week or twice a week. And if you do that in the context of chemotherapy, again, mitoxantron, and this is a model of fibrosarcoma in immunocompetent animals, you see that when you starve your animals plus mitoxantron, then you get a better effect of the treatment. Same with oxaliplatin, which is another drug that induces immunogenic cell death. And this depends on the immune system, because if you do it in immunodeficient animals, now you get nothing. It's not a trophic effect. It's not. It's an immune system dependent effect. Starvation is always seen in a bad way by clinicians, and they probably have a point. This is what happens with the mice. If you starve them for 24 hours, they lose 10% of the weight. If you starve them for 48, they lose 20% of the weight. If you starve them for 72, they die. But if you think about it in human terms, this is impossible. I mean, if you starve yourself for 24 hours, worst case scenario, you're going to lose half a kilo if you're lucky, or probably not even that. But anyway, we shifted very quickly our attention from starvation to molecules that have the same biochemical effects of starvation. Notably, they drive autophagy, but they do not induce weight loss. And those are, we call them caloric restriction mimetics. They include resveratrol, ripamycin, spermidine, and many others. You see here, when you give them to the mice in a continuous manner, the mice do not lose weight as compared to the massive weight loss that you get with starvation. So we repeated the experiment I showed you before with these drugs instead of starvation. Again, mitoxantron alone in blue, and then the combination with the different caloric restriction mimetic always gets a better response. And it always goes away when you do it in a nude animal. So it means also in this case, it's dependent on the immune system. Same thing with another model. I don't want to go through that. Conclusion, second round of conclusion. The espresso approach is good. Actually, it's not that simple. And I learned that on my skin when I moved in 2017 at radiation oncology here at Cornell. I really wanted the Starbucks approach to be bad and the espresso approach to be good. So I started to test it in our own systems here, and I found another degree of complication. Why is it so complicated? Because I mentioned before, autophagy in cancer cell here, it's important for the cancer cell itself to save cancer cells from stress. It also generates signals that are going to the tumor microenvironment, and they're talking to same cells that are not undergoing autophagy, to different cells that are not undergoing autophagy. For instance, the immune system. I told you before, ATP is released in the microenvironment because of autophagy, and the immune system is detecting it. Also, autophagy can drive the secretion of hormones that go in the circulation, and they talk to distant cells, really distant cells. And this is work from a late friend, Beth Levine. She demonstrated in another field, in the field of metabolism, how, for instance, autophagy in the muscle regulate glucose homeostasis in the liver through the secretion of hormone that go in the circulation. So when you move to a radiation oncology department, the first thing that they tell you is that there's this thing which is called abscopal response, and they really consider it as the holy grail of radiation. What does it mean? Well, it's a complicated word to say to define a very rare clinical event, which is you have a patient. The patient has many tumors, like this patient here. You see all these dots are tumors, and you irradiate one specific tumor for a reason. Maybe in this case, it was a palliative radiation to a liver problem. This patient received radiation only to that lesion, and look what happened. Magic, all the lesion went away, and that's called an abscopal response. You see, this was a year later, and the patient is still alive today with this kind of disease. You can call it a miracle, or you can call it abscopal response, but basically it's nothing short of a miracle. Data from my department prior to my joining the department, and specifically from Sandra De Maria, demonstrated in mice that the abscopal response is an immunological effect. How did they do that? They implanted two tumors in the mouse, one in one flank, one in the other flank, and they irradiated just one. And they noticed that when they irradiated one in the context of an immunotherapy that per se is completely inefficient, they could get the control of the non-irradiated tumor. That was not happening in immunodeficient animals at all. You see, the irradiated tumor is responding, but the non-irradiated tumor is absolutely not responding. And it was not happening when the non-irradiated tumor was a different tumor. So not only it's an immune effect, but it's an antigen-specific immune effect. And they also went on to demonstrate that this antigen-specific immune effect is due to the fact that when you irradiate cancer cells, they accumulate DNA in the cytosol. And that DNA is important because it drives the secretion of these DAMPs, of these cytokines, that then recruit immune cells to mount an immune response. Specifically, they drive the secretion of type 1 interferon. Critical work from Claire Van Puy and Sandra De Maria in the 2017-18. So we knew from the literature that autophagy is a very potent inhibitor of type 1 interferon signaling in many other fields. Infection, like direct permeabilization of the mitochondria, but there was kind of a solid consensus about that. But nobody tested what happens with when you use radiation, and what is the impact of autophagy in the abscopal response to radiation. Knowing that radiation is a very good immunogenic cell death inducer. So our expectation when I joined this department was autophagy is going to be needed for immunogenic cell death driven by radiation, exactly as it's needed for immunogenic cell death driven by chemotherapy, which I showed you before with mitoxantrone and oxaliplatin. And this largely work of Taka, an exceptional postdoc of mine, and Marisa, who was visiting the lab as a medical fellow. So what we did first to study that, first we wanted to have the tools. So we generated autophagy deficient cells. In this case, we're talking about mouse TSA cells. It's a model of hormone receptor positive mammary carcinoma. We did ATG5 deficient cells. and you see ATG5 deficient cells, they lack the ATG5-12 conjugate, which is normal. They also lack monomeric ATG5 here. We also did ATG7 deficient cells. ATG7 deficient cells, of course, lack ATG7, which is expected. They lack the ATG5-12 complex because ATG7 is responsible for the formation of the complex. But of course, they do not lack ATG5, which in this case, it's then monomeric. When they are wild type, all the ATG5 you have, it's in the complex. So now we have ATG5 deficient cells and ATG5 deficient cells, ATG7 deficient cells. When you do irradiate your cells and when you use starvation as a positive control for autophagy induction, you see a classical Western blot for autophagy induction, which is as follows. P62 is an autophagy substrate. It gets degraded when you starve your cells. It gets degraded when you irradiate your cells. So it's aligned to the fact that autophagy is activated. And all of that can be inhibited by a lysosomal blocker, which is bacillomycin A1, which is also consistent with autophagy. So indeed, autophagy is defined by the lysosomal involvement. So now we degrade this by starvation, we degrade this by radiation, and it's recovered when we combine that with lysosomal inhibitors. So this is a proof that radiation is actually driving autophagy. Nothing of that happens when the cells are ATG5 knockout or ATG7 knockout, as we would expect. So we started to check the viability of the cells after radiation. And I mentioned before, we really went the solid way, which is, can cell pick up PI, which is this vital dimension or not? So basically, can cells become permeabilized after radiation? We give radiation at 2 different doses to control cells and cells that lack ATG5 or ATG7. And you see here, the percentage of cells picking up porphyrium iodide after radiation is much higher when they lack ATG7 or ATG5, which is expected because autophagy is there to save the cells. Same thing when you did clonogenic assays, which is a technique that radiation oncology love. And in this case, again, this is the clonogenic survival upon radiation of control cells. And this is way decreased when the cells lack ATG5 or ATG7, no surprise. Again, here started the surprise, and we were really puzzled about it. Why? Because I mentioned before, when autophagy deficient cells are used to vaccinate mice, and those cells are treated with chemotherapy, they lose the capacity to vaccinate the animal. I also mentioned radiation therapy is a very potent immunogenic cell death inducer, and indeed, when you vaccinate mice with irradiated cells, all the mice are protected from another challenge later on with living cells of the same type. Very surprisingly to us, the capacity of radiation to drive immunogenic cell death was not compromised at all by the lack of ATG5 or ATG7 as compared to here, when the capacity of chemotherapy to drive immunogenic cell death was completely compromised. Also, when we went in vivo in immunocompetent animals, radiation was a good controller of the disease. When we combined radiation with a autophagy inhibitor that we would expect to make this worse, actually it made it better. The control of the disease here by an autophagy inhibitor is better than the control of radiation alone. So we started to think, okay, there's something going on here. And we repeated the same experiment with ATG5 knockout and ATG7 knockout cells, and we got the same results. Radiation is better in vivo in immunocompetent animals. No, we're not talking about immunodeficient animals as in the case of chemotherapy. No, this is an immunocompetent animal. It's better than radiation alone. So autophagy inhibition ameliorates the efficacy of radiation. So since we had this preliminary data and literature about type I interferon responses, we started to check for type I interferon. And indeed, both in the case of chemical inhibition of autophagy and in the case of genetic inhibition of autophagy, we were able to detect more type I interferon, both as a transcription of the gene. You see here the response to radiation alone. This is the response to radiation in control condition, much better when you inhibit autophagy, also here. And this is the same data with ELISA. You see the increase that you obtain when you combine pharmacological or genetic inhibition of autophagy to radiation only. We demonstrated this is dependent on cGAS and STIG signaling, which means it depends on cytosolic DNA. when we downregulated cGAS or STING by using a doxycycline inducible construct, then we lost not only the effect of radiation alone, but also the beneficial effect of combining radiation with pharmacological autophagy inhibitors. And you see it very nicely here. And also we tested a chemical cGAS inhibitor here, but it's not, just to demonstrate it's the same in the knockout cells. Of course, we knew from Santos de Maria data that this is due to the accumulation of cytosolic DNA. Also, we just proved that it's dependent on cGAS, which is a cytosolic DNA sensor. So we wanted to quantify cytosolic DNA, and for that aim, we developed an immunofluorescence approach that does not permeabilize nuclei. So it basically only stains double-stranded DNA molecules that are available in the cytosol. So the nuclear membrane and the inner mitochondrial membrane are perfectly intact by this permeabilization approach. Otherwise, as you imagine, you would see a very bright green nucleus, which we don't see. And again, we can use automated image quantification here to do the same trick. And you see radiation elicits accumulation of cytosolic DNA. This is boosted when we combine chloroquine, and it's boosted in ATG5 knockout and ATG7 knockout cells. And this was in line with data from the Rockefeller Institute. This was in line potentially with data from Memorial, and another team demonstrated that when you irradiate the cells, you get cytosolic DNA accumulation through micronuclei. Also, we knew that micronuclei are a very good substrate for autophagic removal. Both work from Guido and from other team. So basically, we hypothesized, okay, what we see in our cells is micronuclei. So we started to go to test that hypothesis by staining irradiated cells with the antibody I told you before for double-stranded dsDNA, which is in red here, and with an antibody that is specific from the nuclear lamina. And we were surprised to see two things. First of all, there was not a lot of micronuclei in our culture. You see here, nuclei look kind of normal. And second thing, the dots were not in the proximity of the nucleus. They were somewhere else in the cells. Of course, what could be the other source of double-stranded DNA? Mitochondria. So we did another staining. In this case, we stained again for this double-stranded DNA species in red. You see that the nucleus remain intact. We remain not permeabilized by the procedure. But now we stain mitochondria with COX-4 antibodies, and you see this very nice co-localization of the signal. We quantified that, demonstrate that most of the cytosolic dsDNA positive dots are in the proximity of COX-4, so mitochondria, and not laminin B, so micronuclear. Also, we stained for TFAM. TFAM is a mitochondrial transcription factor that is associated with mitochondrial DNA. And indeed, when we irradiate the cells, you see these very nice yellow dots, showing that the majority of dsDNA that is found in the cytosol of irradiated cells colocalizes with TFAM. And this is the quantification of that. We wanted, that is observational data. We observed double-stranded DNA in the proximity of mitochondria. So we hypothesized that mitochondria were the source of that DNA, but we didn't prove it yet. It could be nuclear DNA going out from the nucleus and just sitting close to mitochondria. To prove that, we generated so-called Rho0 cells. Rho0 cells are depleted in mitochondrial DNA. As you can see here, there's this very faint mitochondrial DNA band. The genomic DNA is actually normal. These cells have a hard time growing in culture. You need to give them a lot of nutrients because they cannot count on mitochondria to do their job, but you can maintain them. And now the interesting part is when you irradiate these cells, now you no longer have the accumulation of double-stranded DNA in the cytosol, which of course means now mechanistically that the cytosolic DNA that we find upon irradiation comes from mitochondria. And you see here very nicely in this staining, it's all gone. This is the irradiated cells, this is the irradiated Rho0 cells, and this is the quantification. Same thing with confocal, not important. This is more important. Now, when we irradiate Rho0 cells, they are no longer able to secrete apointerferon because they don't accumulate cytosolic DNA upon radiation. And there was a paper from an Australian group demonstrated that when mitochondrial DNA is released, it's not really released, but many times it sticks to the mitochondria, as in see here, these guys use super resolution microscopy, and they also demonstrate this is a back apoptotic pore dependent mechanism. And if you look here and we go back quickly to this slide, you will note how indeed we were not saying real colocalization, we were seeing like juxtaposition between the green mitochondrial signal and the red. You see here, they really look next to each other, which is similar to what these people here were finding. And again, they said the back are important, so we tested that. We tested in two ways. First of all, we overexpressed BCL2, which is a back inhibitor. And indeed, we were able to reduce the amount of cytosolic DNA and the amount of type I interferon that we observe upon irradiation. This was a marginal effect, I would say, because TSA cells, they're actually BCL2 competent. So BCL2 is already there to give the job. We overexpress it, we get something better, but not that better. So we took another model. This is thanks to the nice gift from Bert Vogelstein of the HCT 116 cells that are either double knockout or they are backs heterozygous, so they have backs. None of them have back. So these are double knockout cells, and you see nicely now when you irradiate the double knockout cells, the release of DNA, mitochondrial DNA, we can now say it's inhibited as it is the point of ion secretion. And this goes in line, was supported by data from Rockefeller, where they actually demonstrated that nuclear DNA is a bad cGAS activator. And it makes sense because imagine if at every mitotic round, your nuclear DNA would activate cGAS because it gets exposed to the cytosol. This is not good. You need protection from that. And one protection is actually given by histones. So it was reassuring to see some other data. We wanted to merge the two. I mean, at that time, we had two stories that were separated. We had the mitochondrial story and we had the autophagy story. But we didn't know whether autophagy was really picking up mitochondria there, what was happening. So we did Rho0 cells in the ATG5 knockout and ATG7 knockout background. And as I told you before, when you do the ATG5 and ATG7 knockout, you get much more dsDNA upon irradiation. But look at this. This is completely abrogated when those cells are Rho0, meaning that indeed the dsDNA that drives this increased response comes from mitochondria, as is the dsDNA that drives the scramble response. This is same data with the pharmacological inhibition of autophagy. If you just focus on the right, you see here the ability of hydroxychloroquine to boost the response given by radiation, and it's gone again when Rho0 cells are on board. We wanted to do two more things, and then I'm gonna stop because I think you guys are bored by now. We wanted to have an in vivo thing, and we wanted to have a human thing. The in vivo thing, we just did what I told you before, which is you generate mice with two tumors, one on one flank, one on the other flank, and you irradiate just one. And you can do that with the cells that you like sometimes. In this case, we were lucky we could do it with our cells. So we did it in a control condition. Both tumors are wild type or control. And in a condition when the irradiated tumor, but not the other tumor, was ATG5 or ATG7 deficient. This is the control condition only, just to explain you what goes on normally. What goes on normally is that this is the irradiated tumor. The part above here, primary tumor, received radiation. And when you irradiate the tumor, the black line with a square and a triangle, of course, you get a response that is, of course, inhibiting tumor growth. When you use a checkpoint blockers, so this is a CTLA4 blocking antibody alone, doesn't do anything. And this is important. Checkpoint alone doesn't do anything. When you combine them, radiation and checkpoint blockers, you get a better control. And now you also start to get eradication of the disease. So now, in this case, 80% of the animals eradicated the irradiated lesion. Remember, this is the irradiated lesion. Now we look at the other lesion in the same animals. When you treat this lesion, the other lesion that is down here has never received the treatment, with the only exception of the systemic checkpoint blocker. But I told you before, systemic checkpoint blockers doesn't do anything. And indeed, it doesn't do anything here either. When you irradiate the other tumor, of course, this tumor doesn't care because you're irradiating the other one. But when you irradiate the other tumor in the context of checkpoint inhibition, now you start to get an immune response that takes care of the non-irradiated tumor. And it's better when you use this 8 gray 3 times schedule. I don't want to go into those details, but now just think we have a situation where we can control a tumor that is not irradiated by irradiating a distant tumor. But also we can control it, but there's no eradication at all. The eradication is only in the side that you irradiate. And again, this is control condition. So we then went for our genotype differences. And in the left, whatever you see in the left, it's not important. It only means that you can make your treatment better at eradicating the tumor that you are irradiating, because on the left is the tumor that you are irradiating when the tumors are ATG5 or ATG7 deficient. But we told that before. It's not important. I mean, it's just making the tumor more sensitive, right? What is important is on the right. The only situation where you now get eradication of the non-irradiated tumors in a percentage of the animals is when the irradiated tumor, the other one on this side of the animal, is ATG5 or ATG7 deficient. So now we have 40% of the animals here that are tumor-free at both sides. Here is the irradiated side. We can make it. It's easy. I'm talking about here. We are making an immune response so strong that it eradicates non-irradiated lesion. And it depends on type 1 interferon, because if you inhibit type 1 interferon with an antibody, as here, this curve, dotted curves here, then you lose that control. You go back to a normal control. It doesn't matter for the primary lesion. You're irradiating that lesion is going to respond. It matters for the lesion that is not irradiated, the lesion that needs an immune response to be controlled. Finally, we went to a public data set to check for some sort of relevance in our findings, right? And they focused on this signature here, which includes the genes we deleted, ATG5 and ATG7, but also other genes. And guess what? When you stratified patients with breast cancer, as I mentioned, all this work was in breast cancer. So when you stratified patients with breast cancer based on their autophagy signature, which we defined as a mean or average of these genes here, those that have a high autophagy goes worse. Those with a low autophagy goes better, but it's completely expected. I mean, autophagy is there to help cancer cells, right? Do we know that it's not new? Okay, not very interesting. What is more interesting is that you can use that autophagy signature to separate the cohort in 2 cohorts, 3 cohorts, or 4 cohorts, depending on where you cut it, right? You can cut the cohort in 50%, 33%, or 25%. And no matter how you cut your cohort, you can then measure other signature in those groups. One of that is type 1 interferon signaling. Now you have patients with a high autophagy, and then type 1 interferon is low. Now it can be median, tertile, quartile, always the same. The more autophagy, the less type 1 interferon, which is in line with our finding. Same thing for interferon gamma, which is a signature of active immunity. The higher autophagy in those patients, the lower signature of activation of an immune response. When you do unbiased, clustering of these patients, you see that you get nice clustering, but the autophagy status, which is up here, doesn't really. There is an enrichment, yeah, but it doesn't really cut it all together. It's not as if all the autophagy high were on the one side, all the autophagy low were on the other side. Still, you can see something. But when you compare autophagy high to autophagy low and you do gene signature, then you see that the most downregulated signature of all, in an unbiased manner, they are interferon gamma and type 1 interferon signaling. So high autophagy, again, low type 1 interferon and interferon gamma signaling. Our story was a story about mitochondria. So we wanted to get a sense of what's happening with mitochondria here. And that was complicated because this is transcriptional data, publicly available. There's no data on mitochondrial DNA. There's no data on mitochondrial RNA because those data were collected with the previous chip-based systems where the chips were based on poly-A tails of messenger RNAs. So mitochondrial RNAs are not polyadenylated, so you cannot capture them on a chip. And again, this was in the pre-RNA-seq data. So if it were RNA-seq data, maybe there would be a signal. But we reasoned, where do most of the mitochondrial proteins come from? They come from the nucleus. So if you have a lot of mitochondria in your cells, you must have a lot of nuclear, now polyadenylated RNAs that code for mitochondrial proteins. So we did a big score, including all nuclear RNAs coding for mitochondrial protein, and we call it MitoScore. Now we look at these patients again based on autophagy signature. The higher autophagy, the lower the mitochondrial abundance in those patients. So now we have a model for this, which is here. The model is that autophagy comes in play up here to take care of permeabilized mitochondria that release mitochondrial DNA. This mitochondrial DNA is very important for cells to drive type I interferon secretion through cGAS, which ultimately generates anti-cancer immunity. And now we proved it in the mice and cells. We proved it step by step. In the patients, we have signatures saying that when autophagy is high, mitochondrial content is low, type I interferon content is low, and activation of cancer immunity is low. Can we target this therapeutically? The answer is autophagy, too preliminary, too complicated. I'm going to talk about it in a second. Caspases are inhibiting this. So maybe we could target caspase, and there is a drug which is available in the clinic, but it's still a bit controversial. I know people don't like the idea to inhibit caspases in cancer therapy because they believe caspases are there to kill the cells. I told you before, caspases are mostly there to regulate the immunological phenotype of cell death and the speed of cell death, at least in humans. But we have these wonderful drugs called venetoclax. Venetoclax is a mitochondrial permeabilizer. So we now can think about combining radiation with this drug to boost the system, to overwhelm autophagy here with too many mitochondria permeabilized so that we can get this better immune response. And there is safety data about venetoclax in breast cancer patients, but this is not important. So 2 questions. First, should we inhibit or activate autophagy? And that is really a complicated question now. When I moved to Cornell, I would answer we should activate autophagy. Now I no longer know about it. What I do know, it's a context-dependent scenario. So if you are in this scenario here, you don't want to go for espresso. Absolutely don't go for it. If you're in this scenario here, you will not even find Starbucks. So you should probably focus on espresso. So you really need to understand what the tumors look like, if the tumor depends on autophagy for survival, if there is an immune response that depends on ATP, mostly as in the case of chemotherapy, or it depends on type I interferon, mostly as in the case of radiation therapy. And also what we are missing here is a very important thing, which is targeted agents. Activating autophagy at the whole body level, it's not bad. You can do it. You can do exercise. You can do intermittent fasting. You can take this caloric restriction remedy. It's not going to be a problem for the rest of your body if you activate autophagy at the whole body level. But inhibiting autophagy at the whole body level, it's bad. Your brain needs autophagy. You cannot think about, okay, now I'm going to give an autophagy inhibitor to a cancer patient for like 6 months in a row. So either we develop a way to target cancer cells for autophagy inhibitors, or we develop very cyclic, very intermittent regimens whereby we inhibit autophagy for a week and then we stop it for 3 weeks, these kind of things. Also, we need better drugs. Lysosomal inhibitors as chloroquine and hydroxychloroquine are not valid and viable for this approach because they inhibit too many things. They inhibit autophagy, yes, but they inhibit phagocytosis, endocytosis. Anything that is vesicular is inhibited by these drugs. So we should go better. So to answer the title of my talk, why so many ways to die, I really think, and I think it's emerging strongly from the literature, that it's all a matter of keeping your body healthy. There are so many ways to die because you need ways to drive an immune response when some type of stress conditions are around, pathogen infection, cancer. You really need that to be detected and picked up by the immune system. And there are mechanisms for that. But most of the time, when your cells are dying because you are an adult and your intestine needs to be replaced and your blood cells need to be replaced and they are going to die, you don't want that. And there are mechanisms of cell death in place for that. Apoptosis is one of those. Most often apoptosis is driving tolerance and immunological silence. Other mechanisms of cell death are more immunogenic, and they come up when it's needed. And that's why we can stay together and not succumb to autoimmunity in our adult life. Most of the work I showed you is from Taka, as I mentioned before. This is my current configuration of my lab. Actually, one person is missing from this slide. We also have a supporter that is very helpful, has been very helpful during the COVID times to keep the morale up, and that's a big boy. And those are all the people who collaborated. I don't have time to go through all that, and the funding agency that support our activity. And with this, I think I talked too much, and I'm happy to take any questions.
Excellent, man. You never talk too much.
Impressive, yeah.
You always say things that are extremely interesting and important.
Of course. Great, great.
Thank you.
So let's now
people, yes,
ask the students to turn the camera on and start ask
the questions.
So many ways to die.
It was the title of the Keystone that we organized in Brazil in 2019. Actually, the last meeting in person that I attended. Why so many ways to die?
And the interesting part is that there's even others coming up right now. There's this cuproptosis that has just been published in Science, and I still are kind of still short of understanding the entire panoply of signals that are available to regulate cell death, which has become, if you think about it, I mean, I always keep this eye on evolution, right? If you think about it, at the very beginning, we were unicellular organisms, right? And we wouldn't really care about supporting the others, or, I mean, it was mostly of a thing. I need to survive because I need to pass my DNA, which is the only place my DNA is, to the progeny. And then we became colonial organisms. So we were still unicellular, but we were aggregating, and already at the level of colonial organisms like yeast, there are mechanisms that the cells take advantage of to sacrifice themselves and give to the others, especially in nutrients, in nutrient shortage. Some of the yeast cells will die, but not all of them. And that's going to be nutrients for the cells that are around. And then we evolved to a situation where we are multicellular, and now we have to keep track of the entire organism. So that's the complication. I think it evolved with that transition from unicellularity to multicellularity.
There's a very nice example of the cooperation in unicellular organisms as well that was published, sometimes back by Marcelo Barcinski, a friend of us here, parasitology, immunologist in Brazil. By chance, I was a collaborator on the paper. But so what we show is that because this particular bug lives inside our organism, so they develop a system where part of the living Leishmania externalize phosphatidylserine. So they are living as a matter of all you can say. So they are with membrane integrity, they can divide, whatever, but then they fool our immune system to believe that this is something to be tolerant. And the Leishmania that does this, so they kind of sacrifice because the immune system deal with that portion, but the other bugs would just take over. And it's amazing, evolutional thing regarding kind of interaction, host-parasite interaction that is not quite apoptosis, although there was externalization. It's not cell death, but uses the
mechanisms,
the mimics, yes.
Yeah, so that's, they say apoptotic, we say apoptotic mimicry.
Mimic.
That was we have Gustavo Martins.
Gustavo Martins, yes.
Can you guys hear me?
Yes.
Yes, perfect.
Okay, so thank you, Professor, for the astonishing lecture. Your presentation was flawless, and just to entertain you and Professor Gustavo and Professor Karina with a little bit of my ignorance, when I first enrolled in this course, in this discipline, I thought that we were going to talk about mainly apoptosis and phagocytosis and autophagy and maybe a little bit of necroptosis. And I have never imagined that cellular death was so large a topic, and that where it was so beautiful, the mechanisms involved, and it was so instigating as a subject of research. And I think that has been a very pleasant surprise of mine. And about my question, of course, during your presentation, and you talked a lot about tumor cells and about the immunogenic cell death elicited in tumors and malignant tumors. And it kept me thinking if it may be also correlated if immunogenic cell death may be also implicated in autoimmune disease.
Yeah.
Kind of.
Yeah, it's a very appropriate question, and the answer is yes. we started to dig about that with a colleague of mine whose name is Laura Santralborgio. She's an expert in proteomics, and she's an expert on antigen presentation. She has not worked a lot with cancer. She has always focused on really other types of pathophysiological settings. And one of the settings that she has been working on is obesity. And actually, as you might know, obesity is associated with a lot of metabolic derangement. So we modeled that by using mice that are fed a high-fat diet, so they get literally fat, and we measure what's going on in their liver, for instance. And we recognize that their liver is subjected to an extraordinary degree of oxidative stress. And that is the cause for a change in antigen presentation. So now the liver is no longer presenting normal antigens that are covered by tolerance, because the thymus did the work and it erased the T cells that would recognize those antigens. But now the T cells that could recognize those antigens are available. So those are modified self-antigens. They're not genetically encoded. The genetically encoded portion is normal, but the oxidative modification changes directly the antigen as a chemical reaction, but also changes the way that the antigen processing machinery is processing the antigen. So the peptides that come out from the same protein, when the protein is reduced versus oxidized, are different. So the cells are exposing different peptide on their membrane, and now they become able to drive an immune response. And it's a very beautiful work that she did, I think, because it raises the attention to the fact that at the end of the day, what counts for these autoimmune and autoreactive disorders is really the availability of CD8, of T cell clones that could drive the disease, right? And of course, you don't expect T cell clones to be available if the antigens that are perfectly normal, because you would have already an autoimmune disorder, right? So you need some changes, and those changes are much less so due to the change in the CD8, in the T cell compartment. They are much more due to non-genetic post-translational and enzymatic reaction that go due to the damage that is imposed by for instance, in this case, obesity. But I wouldn't be surprised to see similar things in many other autoimmune situations. And obesity is not considered an autoimmune disorder, but there is an inflammatory component. It's mostly an autoreactive disorder. So there is inflammation, and inflammation aggravates it. So, but that's, so the short answer to that long one is, yeah, absolutely. And I think there are, even in cancer, those antigens are way less considered than they should be. Now everybody is looking for mutational burden. First of all, mutational burden doesn't tell you if the antigen is really presented because it's just a genetic information. Maybe the gene is not even expressed in cancer cells. We don't know about that. But even so, it's really, even the people who do it in a more precise manner, so they go for after the antigen themselves, normally tend not to consider these post-translational modifications that are important.
Lorenzo, can I just jump into this autoimmune responses pathways? Because there's something that puzzles me a little bit, is that the majority of our autoimmune diseases has a very strong B cell compartment, that lots of antibody, and we always have this thing in mind that maybe it's a cross reaction through some bugs, some pathogens. But now when you show, this immunogenic cell death that may not related to infections, it can be only like cancer, stuff like that. So how would you put this together? So would you say that these dendritic cells will pick up and be activated by DAMPs, and then activated CD4, and then the CD4 will collaborate? Because it appears to me that the B cell compartment is less responsive, let's say, to the DAMPs, but maybe I'm wrong. So, which make then the diseases so hard is not only, as you say, that you need to get availability of the T cells, but also B cells recognizing this post-translational modification, something like that. How do you see that?
Yeah, optimal B cell responses, as you know, need the help and help is CD4, right? And that's exactly what you mentioned. I believe there is a collaboration, and that's, I believe, one of the key scenario where this happens in the cancer setting is tertiary lymphoid structures. So I mentioned before lymph nodes because it's a classical view. The dendritic cells pick up their antigens in the tumor microenvironment. They migrate to tumor draining lymph nodes where they encounter the T cells and the primary immune response. But there's accumulating data, clinical data and preclinical data, demonstrating that the tumors that contain a higher proportion of tertiary lymphoid structure are those that respond better to a wide panel of intervention. And tertiary lymphoid structure are basically, I don't know how to call it, archaic or like simpler version of lymph node where there's dendritic cells, where there's B cells, and where there's this T cell component. So, and going back to the pathogens, I mean, immunogenic that also happens after pathogen infection, and there the source of the disease is very clear, right? If you think about it, and it also again reflects the evolution of the immune system, the overall concept of viral mimicry for cancer cells is very important. Why? Because at the end of the day, the antigens that are going to drive an immune response will be, we can be positioned at different distance of similarity as compared to our own antigens, right? They can be our own, but they can be modified or they can be mutated, whatever. I think the more this mutation and this modification renders them structurally similar to viral antigens and pathogen antigens, which are really different from ours, the more their response is going to be strong. And most of the mechanisms that are in place for immunogenic cell death to cancer evolved as part of immunogenic cell death to pathogens. Type 1 interferon, which is critical for immunogenic cell, is the antiviral mechanisms of all, way before being the mechanism that attracts dendritic cells. No, just shutting down neighboring cells to viral persistence. So there is, I think there's two things evolved together, and now we can count on this system where modification of our own antigens in the context of the right danger signaling drives drives immunity. Going back to the T cell compartment, I didn't mention it before, but the study we did with the liver and the obesity, yeah, we have a B cell clones that produce a pathogenic autoantibody that we can take from a mouse, put in another mouse that is also under a high fat diet, and we can aggravate the pathology of the other mouse by taking an anti-antibody, which is specific for an oxidized variant of, by the way, ERP57, which is one of the stamps.
That's amazing. That's really cool.
Yeah.
Yes,
I mean, the manuscript is under revision now. So I mean, we are going to resubmit it, I think, in a month or something similar. Let's see whether the reviewers will be happy.
Looking forward to.
very nice.
It's amazing because the story of the virus and
Tumors,
cancer, because and the interferon type 1, as you said, because you show in a different context, you know, the importance of detection of the mitochondrial DNA, and it's mitochondrial DNA. So you're not talking about the mitochondrial proteins that are coded by the genome. So it is actually the mitochondria, which is, you know, this ancient bug that invade our cells.
Yeah.
And the other thing I remember that we discussed it last year was the kind of a similar idea is the paper that come from Daniel de Carvalho's lab, my former PhD students, showing that the genetic modulators facilitates cancer immunogenicity by reactivating the endogenous retrovirus.
Yes.
And now cohabitate our genome. So, I mean, the story of tumor immunogenicity will come to, you know, whatever was previously infected in our ancient story. So this is, it's incredible.
I think we were, as a eukaryotic organism, we were really good at harnessing these entities towards our own benefit. And actually, as you met mitochondria, we just talked about mitochondrial DNA and cGAS, but there's way more than that. If you think about formulated peptides that are immunostimulatory, if you think about cardiolipin that is immunostimulatory, so mitochondria are basically a bag of danger signals that we can keep together, and when it's needed, we can release them. And there's even the structural organization of mitochondria that have two membranes. It's like a way to preserve the rest of the cell to detect potentially immunostimulatory factors like cardiolipin. Cardiolipin is the inner membrane. If there was just one membrane, cardiolipin would be there, right? It's and even ATP and ROS. ATP and ROS are outside of the cell. They're immunostimulatory, inflammatory.
Oh, Lorenzo, in that same line, because of DNA, mitochondrial DNA and cardiolipins and so on, ROS, blah, 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 are also strong activators of inflammasomes. And we know that autophagy can also inhibit inflammasome activation. Do you look at the cytokine related to the inflammasome activation in this immunogenic cell death in the context of tumor cells?
Yes, we did at the very beginning, and this is work from the Cromer Times, actually the early Cromer Times, I think 2007, 2008, and we published a Nature Medicine paper demonstrating that the way that ATP drives immunostimulation in the context of immunogenic cell death is dual. One is because ATP is a good chemotactic agent, and it can bring in dendritic cell precursors through the P2Y2 receptor. But also it's an immunostimulatory agent through the P2RX7 receptor, and P2RX7
and NLRP3
directed to an NLRP3. And in that case, there is interleukin-1 beta, interleukin-18 secretion. And when you block either of them, you block the cycle of immunogenicity If you do it in an LRP3 deficient setting, you block the cycle of immunogenicity. We had also a role for gamma delta T cells back then in our setting. So it was the first commerce, it was with a Th17 type of response. I think that has been a little bit overlooked lately. I don't know anybody working on that aspect. I mean, we work on gamma delta T cells for a completely different reason. But yeah, inflammasome is very important. No doubt about it.
Yes, and this crosstalk between autophagy and inflammasome are also intriguing because it's clear that autophagy can inhibit inflammasome activation, but inflammasomes can inhibit or activate autophagy. It depends on the cells
it's
and
nice
the bugs.
It's nice because I'm writing a Nature Review Immunology article called Mitochondrial Control of Inflammation, and figure 3 is exactly what you said. And my interpretation of it is when you have limited mitochondrial permeabilization, you have limited signaling through the inflammasome and to cGAS. That is probably promoting autophagy as a way to feed back inhibition and to shut down the signal and just to recover cellular homeostasis. On the contrary, when you have massive permeabilization of mitochondria, massive cGAS and inflammasome signaling, then you want to inhibit autophagy. So now you have a feed-forward circle where you boost it even more, and then you go for massive inflammation. I think that is it's like a rheostat. If you are below a certain threshold, it's going to be a self-containing system, and if you have above a certain threshold, it's going to be a self-amplifying system for inflammation.
Yes,
that's beautiful.
Interesting.
Looking forward as well to read this review.
This is going to be faster, I believe. We're going to submit it next week. So if everything goes well, by the summer it's going to be out.
Send us. Yes.
Send us the pre-form.
Yes.
Well, I can send you actually the submitted version.
Cool.
Okay.
Confidentially, but I'm sure you're gonna.
Of course.
Nice.
Cheers. All right, guys. Any more questions? I think they had a very big day. It's a very exhausting day. They're a little bit
tired.
Yeah.
Tired.
It's perfectly normal. No worries.
So,
but that was really awesome. Oh, Alexandra. Ale.
Clap. Yes.
Oh, this is a clap. Okay, I thought it was a hand.
Clap. Yes. Congratulations.
Thank you very much, guys. I hope to see you soon. Hopefully over there.
Yeah, definitely.
Yes, definitely.
We should work. Karina and I, we get some friends together in this boat and then organize something here. Kind of, we have something called the school FAPESP School of Advanced School, and this is a state agency, funding agency that then provided some money to bring speakers for a big crowd, which then become like a small crowd, like an intensive 1 week course emerge in something. So we'll think about it. We are also kind of tired, but it's so nice afterwards that it's
it's a lot of work, but then it's rewarding.
It's rewarding. It's very rewarding. Yeah, we were talking about it.
Of course.
Anyway, Lorenzo.
So thank you, Lorenzo.
Prego.
And see you soon. Let's keep in touch.
See you soon, guys. Bye.
Okay, guys.
See you.
Bye-bye. Thank you.
Thank you.